Hello, everyone. My name is Jun-Sun Lee. I'm a computational biologist at Michael Smith's laboratory at University of British Columbia. I would like to thank Brig, first of all, for this wonderful opportunity for me to present my research. And it's such a privilege for me to present this in front of a wonderful scientist in Korea and in Korean community. So my talk today is about how complementation of gene expression in presence and absence variation can contribute to heterosis and drought resistance in crop plants, using sunflower as a model. As many of you are aware, the world is suffering from the climate change, and the impact is great on crop production. And many scientists estimate that we need to double the crop production in order to feed the people around the world by 2050. But here are the four major crops that may go through the change in potential average yield for corn, potatoes, rice, and wheat in 2050. And many parts of the area that you can see that there's a dramatic decrease of the yield change, especially the corn and the potato and rice. They do see the significant decrease of the crop production. And as a crop scientist, as a plant scientist, we feel responsible to help and develop the crops that can have a higher yield to be able to meet the needs. So over the past century, a number of the world's most important crops have transitioned from open pollinated varieties to hybrid production. And heterosis, or is known as a hybrid vigor, is the increase in biomass, stature, fertility, and other characteristics that impart superior performance in F1 hybrid progeny over the genetically diverse parents. So here's an example of corn that you can see on the side, the parental lines, and the F1 hybrid is in the middle, and you will see the much bigger the plant height and the production is much greater in the F1 hybrid. However, the less widely recognized benefit has been the impact of heterosis on environmental yield stability, especially in response to various abiotic stresses. So despite its importance in plant breeding and evolution, the genetic, physiological, and molecular basis of heterosis are not fully understood. Here, the three mechanisms of heterosis are widely accepted. The first one is a dominance model. So dominance model has the dominant allele of big A and big B, and that suppress or complement the recessive allele, small a or small b. And this can consequently have the higher performance in F1 hybrid. And this is most well-known mechanism for the heterosis. The second mechanism here is proposed is the overdominance model. This one has a heterozygosity of the B1 and B2 that at the key locus that contribute to heterosis, leading to superior performance. And the last one is called epistasis model. So the epistasis model has a non-allelic gene such as A2 and B1 or A1 to B2 inherited from the parental lines that interact and contribute to heterosis. So the experimental design, such as a quantitative trait locus, such as QTL, or the genome-wide association study, GWAS study, have offered support for all these three models, but imply that the dominance model is most common of all. We also have to recognize that there is a trade-off between the stress responsive genes and the growth genes. So here's an abiotic stress resistance trade-off in heterosis. And you can see that in this model that as we focusing on the growth of related genes, that you may have the expense of the expression of the stress responsive genes. And this can make the crop less tolerant as we focus on their growth. And here's the concept in the structure variation of the copy number variation and the presence and absence variation in the crop genome. So in this case, maize MO17 versus B73, the color coding indicates the type of segment, the position of the centromere are shown here in the middle as a black box. And in maize, the numerous genes with missing expression in one or the other parental genotypes are expressed in hybrids, and the hybrid express many more genes overall than either parent. So although it's not directly linked to heterotic phenotypes, such a pattern of expression complementation in hybrids is consistent with the dominance model, and it is likely resulting from presence and absence variation under dominance model. So we, in this experiment, we focus on the drought tolerance of crop plant. And here are the three mechanisms of drought tolerance. So the plants resist drought tolerance through drought escape and drought avoidance and then tolerance strategy. So the first one is drought escape, and this is well found in the annual life cycle. And it is associated with the phenotype of the rapid growth, earlier flowering, and high photosynthetic capacity. The second one is drought avoidance, and this one has a life cycle of annual and biennial and perennial. And this has associated phenotype of high water use efficiency, low stomatal conductance, and dense and or succulent leaves, and it can have a high root to shoot ratio. Last but not least, it can have a drought tolerance, and it can be found in biennial and the perennial plants. And you can see the osmotic adjustment, accumulation of sugars, and root growth. So now we've focused on the transcriptome analysis, so transcriptomic response to drought tolerance. So understanding the genetics and physiology of organisms' response to abiotic stress can give insights into how they may be modified to better avoid or tolerate such stresses. And I studied transcriptional, which is gene expression, and post-transcriptional alternate splicing patterns that are responding to the responses of the crop plants to abiotic stresses. So here's a schematic representation of gene expression response in abiotic stress. So as the plant recognizes stress, such as salt, heat, cold, drought, or flooding stress, and it can have the signal transduction through secondary messenger, and that can trigger the transcriptional control by interacting with transcription factor. And this can lead to either gene activation or gene repression that are related to the stress response and the tolerance. And that can have the phenotype of the stress adaptation. In gene expression, we can also look at non-additive gene expression patterns. So what it means is that in this model, you can see the low parent of gene expression. Let's say expression level is 2, and then this parent has an expression level of 4 of such gene of responding to abiotic stress tolerance. And you may expect that there's additive pattern, so which will be right in the middle of a high parent and a low parent. But in a lot of cases, you can also see the non-additive patterns, such as it can be low parent-like or even below low parent, or you can have a high parent-like or above high parent expression patterns. I also focus on the alternate splicing, so we can determine differentially spliced genes by calculating the ratio of the alternate spliced isoforms compared to the constitutive form. So as you know that in the alternate splicing, the introns are being spliced out and exons are put together and they're included in the transcription. However, you can have the alternate donor side, which is coming from the 5' end side, or you can have alternate acceptor side, which comes from the 3' end, or you can have from both 5' end and 3' end, which is alternate position. You can also have the exon skipping. So exons are usually included in transcription, but in this case, the exon is being excluded, or you can have the intron being included in the transcription. So this is post-transcriptional process, and interestingly, the parents, the plants have a majority of the alternate splicing patterns in intron retention, whereas in animals, exon skippings are more found compared to plants. And this can generate multiple protein isoforms, and it can be considered as a fine-tuning gene expression, as having intron and such can have a nonsense mediated decay. And alternate splicing is believed to play a critical role in helping plants adapt to their environment, and it can increase our understanding of plants and crop phenotypes. Another interesting fact is the plants possess nearly double the number of SR proteins, serine, arginine-rich protein. They're known for alternate splicing regulator as compared to non-photosynthetic synthetic organisms. Here's some examples that alternate splicing can lead to possible functional mechanism. So the first one is FCA gene. So FCA gene in Arabidopsis is found to be controlling the transition from vegetative to the reproductive phase. And the FCA intronless transgenes can result in increased FCA protein level and accelerate the flowering. So in this case, alternate splicing is preventing precocious flowering, so it's controlling the timing of the flowering. And in Drosophila has about 15,000 genes in the genome, but in this gene called DSCAM gene, because there's so many alternative splice sites, and it generates 38,000 isoforms involved in phagocytosis from this one gene alone. And you can imagine how dynamic this alternate splicing is for this gene. Now we go back to our crop model. So we use a sunflower in this case. Sunflower is a hybrid crop, and we use well-characterized maternal and paternal lines, and they're F1 hybrid. And it's found to be here, so XRQ is a maternal line, PSC is a paternal line, NAD is F1 hybrid. And both control and drought, they were found to have a much higher performance in plant height in F1 hybrid compared to the parents. And this is surprising because higher growth rate and biomass accumulation, you would expect that they require greater water usage. So it's interesting that still have this component of the heterosis under drought condition. So our major questions are there similarity in how sunflowers respond to drought and hybridization at the transcriptome level for gene expression and alternate splicing? And do such similarities offer any clues regarding how heterosis might contribute to drought tolerance. And is alternate splicing is a part of the heterotic response to drought? And lastly, is there evidence of expression complementation of gene presence and absence variation in the F1 hybrid as predicted by the dominance model for heterosis? So these are the questions that we try to answer. So as for our materials and methods, as I mentioned previously, we use the three well-characterized lines, XRQ as a maternal, and PSCA as a paternal line, and NAD as F1 hybrid. And we use this method called FTSW, so fraction of transpirable soil water. So you can measure the water potential of each plant in the pot that's planted in, and you can see if you can measure if it's well water has the greatest this FTSW value will be shown as 1, and the least water or more like a drought condition will be 0 or 0.1. So you can see the gradient of the drought stress that plants are going through, and we collected samples for transcriptome analysis. So we generated 1.8 billion reads from 36 RNA-seq libraries under both control and drought treatments. We also did the phenotyping and genotyping of the sunflower association mapping, as known as a SAM population. So the SAM population includes 288 sunflower cultivars that represents about 90% of the allelic diversity in the cultivated sunflower gene pool. And the phenotyping was conducted in 3 different locations, Georgia, Iowa, and British Columbia, using 271 lines that were available at that time. And we also did the literature review on the phenotypic data for hybrid and the parental sunflower lines in order to determine which traits are likely to display heterotic responses. So going through those characteristics, especially in the reproductive stage, we found the plant height and the plant head weight has the strongest correlation with the heterotic traits, and biomass, stem weight, and stem diameter, they are moderately related to heterosis trait. We also use this platform is called the HeliaFan. So we collaborated with a government institution in France, INHA, that's in Toulouse in France. So I worked there for 2 summers, working with this phenotyping, high throughput phenotyping platform. So this platform helps to grow the plants, but also control and measure these drought stress treatments by watering the plant for the right time, for the right amount, and taking lots of pictures and measurement to keep phenotyping these plants. So here's an overview of my transcriptome analysis pipeline for RNA sequencing. So as we generate the FASTQ files, the star aligner that we use for aligning the reads, and we generate is called the dot splice files. So dot splice file can split to for the gene expression model with the read counts, but also it can generate the alternate splicing ratio. And we use the EdgeR for differential expression analysis. And in-house R script, we analyze the alternate splicing ratio to determine the differential splice genes. So in terms of the presence and absence variation of gene expression, we found 113 genes that were missing in one or the other parental lines that show the expression complementation in hybrids. And we refer to these as candidate genes. And many candidate presence-absence variation genes were expressed consistently in both drought and control treatments. So this offers a straightforward explanation for heterosis and drought tolerance that it maintains through the stress treatments. So as we did the GWAS analysis, and this figure shows the proportion of presence and absence variation regions with the positive or negative relationship on phenotypic traits, and only gene with the presence absence variation with expression complementation in F1 hybrids are included here. And we found thousands of presence and absence variations were detected, and at most, such loci, the absence allele at gene presence absence variations was associated with reduced value of the heterosis related traits, but not quality traits. So this pattern was strengthened for the specific presence and absence variations that showed expression complementation in the F1 hybrid. So we see that the GWAS data with the first top one shows all presence absence variation, and the second one shows the only presence absence variation that differ between XRQ and PSCA, and show expression complementation in a D. And as you can see that you will see the more the heterotic traits as you go, this the positive presence absence variation correlation gets stronger and stronger in all three different common garden experiment. And with the genomic prediction, we used the genomic prediction model was trained with the mixed model, mixed solve function in R package. And this can estimate which traits can be strongly related to those presence absence variation alleles. And we found those the most heterotic traits, the height and stem diameter, they had the strongest, the positive correlation compared to the other traits. And we also investigated a drought-responsive, non-additive expression and alternate splicing events in parental and hybrid sunflower and cultivars. So we found those stress-responsive genes, they're found to be having more upregulation in the F1 hybrid compared to parents. But there was a downregulation of the unnecessary metabolic related genes. And this was highly correlated with the alternate splicing as well. There was a much higher alternate splicing ratio shown in the metabolic pathway related genes, whereas in the stress responsive genes, alternate splicing ratio went down. So this shows the reverse correlation for reinforce the gene expression for this alternate splicing related to the stress responsive genes and unnecessary metabolic pathways. So we propose that there are two molecular mechanisms for heterosis in the crop plants. So the first one here, we found the gene presence and absence variation in parental genomes. So here are the genes being absent in parental genomes. In F1 hybrid, these can be complemented with the expression and complementation in F1 hybrid. So this can strengthen many of the stress-related genes or growth-related genes for both heterosis and drought stress tolerance. And the second molecular mechanism we propose is that we can see the upregulation of the water stress responsive expression and downregulation of the metabolic pathway. and there was a negative correlation between alternate splicing and expression levels. So our result implied that complementation and expression of missing genes in hybrid is a main cause of heterosis in sunflower. And this mechanism also accounts for the yield stability across the different environments, and it can have the second molecular mechanism to fine-tuning those expression to have the drought stress tolerance. So in summary, absence allele at gene presence and absence variation was disproportionately associated with reduced values of heterosis-related traits, but not in quality traits. Gene presence and absence variation differentiating the parental lines that were complemented in the F1 hybrid that's consistent with the dominance model of heterosis. And many of the presence and absence variations were expressed consistently in both control and drought stress, possibly responsible for the maintenance of the heterosis under drought stress. And we found that the transgressively expressed and differentially spliced genes in F1 hybrid compared to its parents that show upregulating stress-responsive genes and downregulating metabolic process to drought. And lastly, the alternate splicing are found to be, their changes are negatively correlated with the expression changes that shows that they can act to reinforce expression differences. So in conclusion of this study, the transcriptomic and GWAS studies of sunflower under abiotic stress show that the hybridization has a profound effect on abiotic stress responses, and the consistency of expression complementation in hybrids in both control and under drought stress that appear to account for the maintenance of the heterosis in different environments as well. But this needs to be explored in a different stress, different experimental setups as well. And shifts in the timing or the different stress level and of expression alternate splicing responses can explain the differences in abiotic stress resistance among the genotypes. And these could not only enlighten crop improvements, but also it can help us to understand the evolution of the resistance in natural populations as well. So we hope to apply this in that area as well. Lastly, I would like to thank my collaborator, Nicolas Langlade and his group that I was working in Toulouse, France for this project. And I would like to thank Keith Adam and Lauren Riesberg for supervising for this projects and all the collaborators who worked with me. And I think I can take questions and answers. Thank you very much.